Welcome back to my channel. So we are going to look at the structure of proteins. So before we proceed, kindly like, share and subscribe to my channel. I really appreciate. So previously we were looking at peptides, and we said that peptides refers to two or more amino acid residues that are linked together by peptide bonds. So peptides are the same as proteins. So in the previous video, we also looked at the formation of peptides. So we are not going to look at it like that in detail. So we also looked at dipeptides. So what are dipeptides? From the word di means two. So a dipeptide is made up of two amino acid residues with one peptide bond. So we gave out an example of dipeptide. We have carnosine and anserine. These are amino acid residues that are found in muscles and also the brain. Then we also looked at tripeptides. So tripeptides are those protein that is made up of three amino acid residues and two peptide bonds. So we also looked at an example of tripeptide, and we mentioned glutathione. We drew the structure of glutathione. So we realized that glutathione is made up of glutamine, cysteine, and glycine residues. So it's also known as gamma glutamyl cysteinyl glycine. We looked at how to draw the structure of a given peptide given specific amino acids. Then we also looked at the role of glutathione. So I won't go into that. We mentioned some important peptides, that is important proteins. We looked at bradykinin, we looked at angiotensin-2, we looked at antidiuretic hormone and oxytocin, which is normally used in the reproductive, that is, it's normally found in the reproductive system. So you will look at that. So in this lecture, we are going to look at the structure of proteins. So if somebody asks you what is a structure, a structure refers to level of organization of a given substance. So in this case, you are going to look at the level of organization of proteins. So proteins have got four different levels of organization. So we have primary level of organization, secondary level of organization, and also tertiary level of organization of proteins, and quaternary level of organization of proteins. So what is primary level of organization of proteins? Primary level, from the word primary level, this refers to linear sequence of amino acid in a given polypeptide chain. So during the formation of primary level of the primary structure of proteins, the amino group, which is at the terminal end, the amino group interacts with the carboxyl group of the first amino acid to form a peptide. So this level of linkage of aminol carboxyl, aminol carboxyl, aminol carboxyl is what we call the primary level of organization of proteins. So let's remind ourselves very fast. So we say from the basic chemistry, we say that a given amino acid has got a central carbon. A central carbon has got the aminol group. A central carbon has got the aminol group. It has the side chain, which is common in the side chain varies in all the amino acid, but the backbone, which is made up of amino acid, the carboxyl group, and the hydrogen atom, are similar in all amino acid. So the backbone of a given amino acid is made up of carboxyl group, the hydrogen atom, and the amino group. The R structure, the R side chain is varied from one amino acid to another. So this first amino acid can interact with the second amino acid through carboxyl and amino group interaction, like that. So we are drawing the second amino acid. So this amino group is bonded to a central carbon of the second amino acid. This is the side chain of the second amino acid. and this is its hydrogen atom, and it should also have the carboxyl group. So let us interact it with another amino acid, the third amino acid, through the amino linkage, like that. I want to write it like this. That is the amino group. It's also bonded to the second to the central carbon again. The functional group. This is the third amino acid, the hydrogen atom, and the carboxyl group, like that. So in this case, I have drawn a tripeptide. So a primary structure of a given amino acid is the linear sequence of a given amino acid, and it also contains polarity. So as you can see, we have the aminol terminal end and the carboxyl terminal end. So by convention, the terminal end that at the beginning of a polypeptide chain is at an alpha aminol group. So this is an alpha aminol group, and the carboxyl group is towards the end of the structure. So each amino acid is a polypeptide. Each amino acid in a polypeptide is referred to as a residue. So a polypeptide chain has repeating units that make up a backbone and side chain which are variable. So we have repeating units. These are amino group, hydrogen atom, and the carboxyl group. Amino group, hydrogen atom, and carboxyl group. These side chains are the ones that differ from one amino acid and another. So to understand about the side chain, kindly go and look at my video of understanding amino acids. I have uploaded that video. So a polypeptide chain have ability to form many hydrogen bonds. So what makes it to have ability to form many hydrogen bonds? It has the carboxyl terminal end and the amino terminal end. So amino group is the hydrogen bond donor. As you can see, this is a H3N positive. So that means it has the ability to lose one hydrogen atom. Then hydrogen bond acceptor is the carboxyl group. So the peptide bonds have got a double bond character. Let me illustrate the peptide bond using a black color, like that. I want to illustrate it using a black color. Like that. So these peptide bonds have got double bond character. So someone may ask himself or herself, what is the meaning of the word double bond character? A double bond character refers to the characteristic of the double bonds that involves lack of rotation. So double bonds do not rotate. So these bonds have got characteristic of double bonds. So apart from these bonds, the rest of the bonds have got ability to rotate around the molecule. So during the rotation of this molecule, we have the interaction of the hydrogen atoms, that is the amino group and the carboxyl groups of the structure. And this leads to formation of a secondary structure of amino acid. So what is a secondary structure? So a secondary structure refers to the twisting of the primary structure into some regular shapes. So these shapes include the alpha helix, the beta pleated sheet, the beta turn, and the omega loop. So let's look at the secondary structure. We are just going to look at the part of the secondary structure. So in the next lecture we shall finish because of the space. So the twists are as a result of regular pattern of hydrogen bonds between amino group and the carboxyl group. So because of the characteristics of the single bonds that are there, this structure is capable of coiling around itself, that is twisting around itself. So this leads to formation of a secondary structure of proteins. So in this case, we have what we call alpha helix. So an alpha helix is a rod-like structure. It has got a backbone on the inner portion of the structure of the polypeptide and the side chain on the outside of the polypeptide. When you look at it, actually you can realize that the backbone is inside, but the side chains, that is the R groups, are on the outside of the structure. So as it twists around, this leads to interaction of the bonds of the amino group and the carboxyl group of the amino acid that is 4 units ahead of the first amino acid. So you realize the amino group will interact with the carboxyl group that is 4 units ahead of it. So in this case, we have the first, I have drawn a nonapeptide that has got 9 amino acids. So the amino group of the first amino acid will interact with the carboxyl group of another amino acid that is 4 units ahead of it. So the process repeats itself, making the structure to be complex and stable. So each amino acid residue uses the amino group to form hydrogen bond with the C double bond O group of the amino acid that is 4 units ahead. So in this case, I have done a right-handed helix which rotates clockwise, while left-handed helix normally rotates anti-clockwise. So in this case, we have this is a right-handed helix. Why do we call it a right-handed helix? It is a right-handed helix because when you look at it, it has a screw sense, so it tends to rotate towards the inside of the board. Look, it goes inside the board, outside the board, inside the board, and outside the board. So a right-handed helix rotates clockwise, while the left-handed helix rotates counterclockwise. So when you look at it, it is rotating like this. So that is it. So let's look at the main features of alpha helix. This one. So the backbone is tightly coiled along the axis of the molecule. So when you look at it, it has a screw sense. So it is tightly coiled around the axis of the molecule. Then the distance between 2 amino acid residues is 1.5 angstroms. So it was found that the distance between 2 amino acid residues in an alpha helix is that amount of angstroms. That is 1.5 angstroms. Then the alpha helix also contains 3.6 amino acid residues per turn. Another important thing is that the pitch of the alpha helix is 5.4 angstroms and the width is 5.0 angstroms. Each peptide bond participates in hydrogen bonding, and this gives maximum stability to alpha helix. Most fibrous proteins contains right-handed alpha helix, which is more stable than left-handed alpha helix. Then the alpha helix is hydrophobic in nature because of intrachain hydrogen bonds. Then the alpha helix forms spontaneously since it is the most stable conformation of a polypeptide chain. So this is the most stable conformation of polypeptide chain. We shall draw other conformation when we are drawing the, that is, we have beta pleated sheet and the beta turn and the alpha, the omega loop, that is, sorry. So alpha helix forms spontaneously since it is the most stable conformation of polypeptide chain. Some amino acids act as terminators for alpha helix, and the contents of alpha helix vary from one protein to another. So in this lecture, we've been looking at the levels of organization of proteins. So we've just looked at one of the parts of the secondary structure of proteins. That's where we have summarized our work. So the secondary structure consists of the alpha helix, the beta pleated sheet, the beta turn, and the omega loop. So we shall continue with the same work. So we've mentioned the primary structure of proteins, and we said that a primary structure of proteins refers to the linear sequence of amino acid in a given polypeptide chain, and it is the simplest level of complexity. As you can see, I have just drawn it as fast as that. So it is the simplest level of complexity. So the major bonds include the peptide bonds, and in disulfide bonds, if any of them are included, and these ones normally come as a result of the nature of the protein. There are some proteins that are made up of sulfur. Examples of such amino acids are tryptophan and cysteine. So if they are included, we shall have the disulfide bonds. So we have also said that the linear sequence of a given amino acid contains polarity. So as you can see, the amino terminal end is positively charged, while the carboxyl terminal end is negatively charged. Then each amino acid in a given molecule of protein is known as a residue. So remember, what is a residue? A residue is substance that is formed after we've given out something else. So this is a byproduct. So this is an amino acid residue, the first amino acid residue, the second amino acid residue, and the third amino acid residue. So that is very, very important for you to know. So you also need to remember that the hydrogen bond donor is the amino group, while the acceptor is the carboxyl group. So you need to remember these features of the secondary structure: the beta pleated sheath, the beta turn, the omega loop, and the alpha helix. Then you also need to remember that the alpha helix is a rod-like structure that is tightly coiled around the axis. Then remember the features of alpha helix. These ones are normally examinable, so you might get a lot of marks here. So remember about the backbone. When you look at it, the backbone is looks like it is tightly coiled around a given axis. It has as crucians. Then the backbone is normally found inside the polypeptide, while the side chain is normally found on the outside, like that, like that. Then during the coiling, the amino group interacts with the carboxyl group, and these ones contribute to stabilization of the structure. So the amino group, which has got a hydrogen bond, that is a hydrogen hydrogen that is a H positively charged, interacts with the carboxyl group. So these ones are what we call hydrogen bonds. So kindly, I want to remind you to like, share, and subscribe to my channel. More and more videos are coming. There are people who have asked me about nucleic acids and so many questions. So I'm going to bring more and more of the videos. So stay tuned, and thank you very much.